Hello and welcome to Pitt Street Research. My name is Stuart Roberts. I'm one of the co-founders of our firm. And joining me on the afternoon of Monday, the 12th of August, 2024 from Sydney is Dr. Steve Gourlay, who's the CEO of Actinogen, ASX, ACW. Steve, good afternoon.
Good afternoon, Stuart.
Well done, Steve. Actinogen has just released some phase 2 data related to your Xanamem drug in major depressive disorder, where those patients also have some cognitive challenges as well. And you've been able to show on an analysis of that data that you're having a pretty interesting underlying impact on a range of measures for these patients.
Yeah, thanks, Stuart. That's true. We're very excited to discover that this drug at a very low 10 milligram dose has a major impact on the underlying biology of depression in the brain, and it has a major antidepressant effect. In fact, the size of the antidepressant effect indicated between sort of 3 and 4 points on this MADRS depression scale, which is used to approve pretty much all antidepressant drugs these days, is totally up there with in the range or even a little bit better than a lot of the approved drugs. So this is a major discovery and validation of the cortisol hypothesis, if you like, that modifying and controlling levels of cortisol in the brain does something that's clinically meaningful.
Right. So Actinogen has been built around this, as you say, the cortisol hypothesis. If we knock down a precursor hypothesis, now I need to check my notes because I always stumble over this. 11-beta-HSD1 is the enzyme that we're knocking down, which then goes on to create too much cortisol in the brain. When I looked at this company about 5 years ago, before you had joined, the evidence was well and truly in from a range of literature going back to the 60s that cortisol tracked closely, excessive cortisol tracked closely with Alzheimer's at least, and subsequently you found it tracks with other things as well. What got this whole program started at the University of Edinburgh back in the day?
Yeah, so as you mentioned, cortisol has been a target of interest in the academic literature for at least 50 years.
Right.
And it's been known to be associated with depression for that whole time. And in fact, there's a test called a dexamethasone suppression test where you give a pill of cortisol essentially and see if it suppresses the body's control of cortisol. And it doesn't in quite a large number of patients with depression. And so So there's been very, very much of interest, but nobody's ever had the right drugs to change this. There is the drug called mifepristone that inhibits the cortisol receptor, and it's a bit too toxic to use as an antidepressant, but it did show some activity in psychotic depression, interestingly, in a small clinical trial. So there was a hint from one interventional trial, at least, that this might actually work. The idea really is that you need your cortisol in times of stress for your sort of fight and flight reaction, but chronic stress in your tissues is bad for you. It basically is metabolically bad. It puts up your insulin resistance, does bad things to your lipid handling and your bone turnover. And in the brain, it seems that it's quite directly toxic and associated with depression and the onset and severity of both of that and Alzheimer's disease. So our drug was designed by the University of Edinburgh chemists specifically to get into the brain, so that at low doses, with a highly potent molecule, we could actually bring down brain cortisol and see if indeed controlling that cortisol had a big clinical effect. Previously, other companies had been going after these drugs as diabetes drugs and designing them not to get into the brain. So this is the first drug of its type that gets into the brain and does what we need it to do.
Right. And we've now tried this out on about 300 different patients. And the effects are pretty encouraging so far. What excites me the most is some news that came in October 2022. You went back to look at the Xanadu, which was the original study in Alzheimer's disease, and then discovered that the patients with whom this drug really worked upregulated a biomarker called, again, I'm checking my notes, p-tau 181 was the biomarker that indicated, and the charts are pretty elegant. The more p-tau, the better our drug works in this situation. That's been a gift for you in terms of crafting up the phase 2B that you're in at the moment.
Correct. So the key thing about early phase or mid-phase development is if you design a really good phase 2A and it's fully informative, you can then design the perfect phase 2B with a high chance of success. And that's what we've done with Alzheimer's disease, particularly with the p-tau analysis you're referring to. And that's what we'll be able to do now with this phase 2A data set from the depression trial, which will be fully informative of how to design what will probably be a longer, bigger phase 2B trial in depression.
Right.
So just to explain that work in Alzheimer's disease, we were lucky enough to have some stored blood samples that were just backup samples that were actually about to be destroyed. And I was lucky enough to discover those were there in time to save them and go back, and we did a new protocol, essentially an in silico protocol with a new a priori statistical plan, double-blind analysis of the new biomarkers that weren't available when the trial was done originally. And sure enough, in people with biologically confirmed Alzheimer's disease with this p-tau level, and it turned we were able to show that in the patients with real Alzheimer's disease, also that was more likely to progress, which is associated with p-tau. We had a big biological effect on the key endpoint the FDA cares about in Alzheimer's, called the CDR sum of boxes. And that was a really big finding. But we also had some beneficial effects in slowing cognitive decline as well in that study. We didn't meet all endpoints, but it was a very, very big clinical effect on CDR sum of boxes, about 5 times bigger than the new antibody from Eli Lilly, for example, that has to be given as an infusion. So if that's repeated in the new clinical study, which is now 36 weeks long, this will be a very, very big new drug for Alzheimer's.
Absolutely. And it's interesting. There was a conference you were presenting at recently when I was in the audience. And that allowed you with a straight face. And I've known you for long enough to know you're a pretty serious guy, Steve. But you're able to talk about potentially this is a 20 or a 50 bagger, obviously subject to some later stage clinical studies based on the market need and the relatively, well, not relatively, the completely new mechanism of action that you've stumbled across here.
Yes, I still think that's true, Stuart. And it's more true today than it was yesterday. These data we just released today are an incredible high level of clinical validation for the mechanism, the dose we're using, and the likelihood of success in future depression and Alzheimer's trials. And so, look, my last job at Principia, I think I put about 400,000 or 500,000 US into the company early at about a $3 stock price, and we've sold it for $100. So that was a 30 bagger. And to be honest with you, I remember saying to myself when I took this job, having done my diligence and found the activity that I suspected was there in that phase 2 study of Alzheimer's, that this should be a bigger opportunity even than that last company, Principia.
Well, absolutely. And just lucky enough to have you. You left San Francisco to come back to Sydney in order to work on this opportunity.
Yeah, well, that's sort of true. I did come back during COVID for family reasons as well. But honestly, when I first looked at Actinogen, I thought doing Alzheimer's disease as a little Australian biotech was crazy talk. But I did spend 3 months of diligence helping Bill Catelby and his team at the time, figure out where to go with their clinical program. And during that time, I was able to look at all of the data, and it was clear that we knew what the dose was. We knew the drug was getting into the brain in inadequate levels. And we also saw signs of life in that Alzheimer's study that I thought were pretty convincing. We were then able to confirm that with a new analysis of that p-tau that we discussed earlier.
Right. So let's talk about today's data, which is very interesting. So obviously we've got parallel tracks here in Alzheimer's and in major depression with cognitive. All the numbers look great on today's analysis. Market didn't like it because the primary endpoint related to cognition. And on that score, that beautiful old thing called the placebo effect seemed to have an effect. The control patients here experienced more cognitive improvement, if I could put it like that, than you were expecting.
That's right. So that was a big surprise because the most cognitive improvement we'd seen in this cog state attention composite was a score of an improvement of 0.15.
Right.
If we'd seen an improvement of 0.15 in the placebo group, we would have been home and hosed with a statistically significant primary endpoint. But instead, we saw an improvement of a whole 0.4, right, which was unheard of and is hitherto unexplained by any cognition experts.
I'm looking out the window of my apartment here and I'm seeing black swans going past the window. It's that sort of outcome, right?
Yeah, it's pretty crazy. So that said, the finding of positive, major clinically significant effects on depression is, arguably more important and more wonderful, because that is saying that this drug is really doing something important to the biologic processes in the brain. Other drugs taken for Alzheimer's and ADHD are sort of revving up, it's like putting more petrol in the tank. You're revving up the neurotransmitters. Antidepressants currently that affect serotonin and dopamine do this. Whereas this is a whole new mechanism where we're probably in the medium term improving the underlying biology of the brain, perhaps by tampering down inflammation associated with cortisol.
Yeah.
And so, it's almost a little bit like the ketamine story, where you give people with treatment-resistant depression ketamine, they can be improved and have breakthroughs that last about a month or so. You still need to repeat treatment, but there is a durable effect from ketamine, which is not due to just revving up neurotransmitters the way that other antidepressants work. So maybe Xanamem is a bit like that.
Right. And this is 100% the layman's approach to this thing, but you talk to patients with major depression, not all of them are having cognitive difficulties. They just feel depressed. So possibly we've got two effects here. If we eliminate the placebo effect we're talking about, we improve depression, and those patients' cognition improve anyway. And so ultimately we'll end up marketing this thing potentially as a depression drug. Is that the thesis we could potentially run with?
I think that's right. At least the top line data that we know today says that this drug is an antidepressant with a novel mechanism and great safety, and could be used in combination or potentially alone as monotherapy, and maybe have durable effects. That's pretty much sounds a bit like a wonder drug for depression. And so I think we will be using depression as the primary endpoint in the larger phase 2b study that's to come next year. And we may or may not choose to measure cognition. I mean, we probably will measure cognition along with a number of other features of depression. But it does look like this drug is really doing something fundamental to the biology and pharmacology of the brain by modifying cortisol. And that's really a sort of a disease-modifying story rather than rev up your neurotransmitters like the other drugs. Right. And so I think this focus on short-term cognition is going to go away, and we're now focusing on long-term benefits in both depressed patients and patients with Alzheimer's disease.
Right. Now, the other interesting thing down the track is potentially we can use that in an orphan condition called Fragile X. Talk to us about that program.
Yeah, so look, the scientific rationale for Fragile X is based on an animal experiment that showed improved behavior in an animal model. That's relatively modest scientific rationale, to be honest with you. And that program has an open IND, so the FDA has approved our phase 2 protocol. But we decided to focus on the adult diseases because we were seeing such strong clinical signals in our healthy older volunteers, and now, of course, Alzheimer's patients, and now in our younger adult depressed patient population. So for right now, that's an important asset. As we go into partnering discussions, I think that asset may be seen as a very interesting and valuable one by certain partners. But for the moment, we're going to stick to our knitting on Alzheimer's disease and depression.
Fair enough. Now, the other thing I like to tell people about Alzheimer's is gradually over time, the bars have been lowered. Not for safety, but in terms of when the agencies are looking for drugs that seem to be doing something, they're getting gradually more reasonable about the kind of endpoints that you can use in pivotal studies. Would you agree with that assessment?
Yeah, I think that's true. The nice thing about Alzheimer's is that there's been a plethora of phase 3 trials agreed with the FDA and the EMA. The endpoints are well understood. The one that we have the biggest impact on so far, at least in our pilot data set, is this CDR sum of boxes. And that is completely risk-free in a sense that from a regulatory point of view. There's no drama about that whatsoever. Right. What's good about that endpoint is it's a global assessment of 6 different domains of how Alzheimer's people are functioning, home and hobbies, social interactions, memory. And it does seem like our drug is making people feel better, certainly making people who are depressed feel better. And it may well sort of have a really good general impact on the well-being of people with Alzheimer's, as well as slowing down their progression. So it is a particularly good endpoint for us. So But there are other validated scales like the one we're using, the Amsterdam Activities of Daily Living Scale, and we're using various validated cognitive measures as well. So we're going to learn a lot from that phase 2B, but it is a confirmatory trial. In other words, we are proposing that, we have the ability to use that trial potentially as one of 2 trials to get the drug registered.
Right. And that's exciting. We were studying this thing at about 10 megs, which is way below the maximum tolerated dose. But obviously, you've got data that shows that's useful. Any unwanted effects at 10 megs, or is this thing perfectly harmless in that respect?
So the new trial showed a very nice safety profile, completely consistent with our other clinical trials at 10 mgs. There was very similar rates of adverse effects. The only serious adverse effect was in the placebo group. There were things like headache that were more common in placebo, and in other things like fatigue, it was equally in both groups. So there were a couple more dropouts due to an adverse event in the Xanamem group. None of those were severe and concerning. So essentially, the drug has an excellent safety profile, and we're nearly up to 400 people treated at this stage. So the safety profile is looking fairly solid going into the larger clinical trials.
Right. Let's talk a bit about you. You're a Sydney boy originally, if I recall. Spent a fair bit of time in California building the company you mentioned called Principia. Tell us a bit more about your background.
Sure. Actually, I'm a Melbourne boy. Melbourne Uni and Monash Uni for my PhD, and Melbourne Uni for my medical training. And then I jointly did a specialty in internal medicine and a PhD with a very famous epidemiologist called John McNeil, and went to UCSF, the University of California in San Francisco in the 90s to do clinical pharmacology, and then decided to take my first biotech job at a little biotech called Genentech in 1997. That was then valued at, I think, $300 or $400 million. And the next 6 or 7 years there was a rich tapestry of learning the ropes of drug development. And
great training ground.
Fortunately, working on so many different drugs that are still used today and were wildly successful. And not everything was successful, I should say, but it was a wonderful learning experience. And of course, the networks I established there then were incredibly helpful for the rest of my career. And I came back and did a stint as a partner with Jeff Brook and Brigitte Smith at GPS Ventures for 8 years, and then went back at the behest of the CEO, Martin, who had worked with me at Genentech, to be the founding chief medical officer of this company, Principia. And we took a couple of small molecules through from preclinical all the way through to phase 2 and phase 3. I'm pleased to say I didn't miss a clinical trial, maybe 10 or 12 clinical trials during that time, including some phase 2s and phase 3s. They were all positive. And that was in immunology, which is a little easier than neurology, I should say. But it was a very good experience taking that company public on the NASDAQ in 2018 and eventually selling it to Sanofi for about a little under $4 billion. So, like I said, I came back to Australia for family reasons initially, but quickly was yearning to get to work again, and assessed a few different Australian companies. And Actinogen stood out as a company with an advanced clinical program that had some missteps, but clearly had a safe and active, interesting looking small molecule. And I still believe that today, obviously, because each time we do a clinical trial, we continue to generate positive data confirming this drug is the real deal.
I must say it's considerably de-risked, I should say, from when I was looking at it in 2018. So well done to you and the rest of the Actinogen team and the board for bringing it this far.
Thanks very much.
Steve Gourlay, thanks for joining Pitt Street Research.
All right, my pleasure. Thank you.